Dr.M.G.R. Educational and Research Institute, University, Madurai, Chennai. Dr.M.G.R. Educational and Research Institute, University, Madurai, Chennai.
It allows behind everything and helps improve lives in a massive way. A very good afternoon, one and all present here. Well, I'm Dr. D. Kavita, Associate Professor from the Department of Chemistry. Shall be host for the day. On behalf of our department and university, I welcome you all for our today's program entitled as Nanomaterial in Antimicrobial Photodynamic Therapy and Biomedical Application by Dr. Busi Siddhartha, Assistant Professor, Department of Microbiology, Pondicherry University. In this studio, we have our department colleagues, Dr. L. Ramapriya, Dr. R. Nitya, Mrs. M.G.R, and Mr. Ganesh Babu for offline support. Now, let's start this program by welcome address. I invite Dr. L. Ramapriya, Assistant Professor in the Department of Chemistry, to give welcome address.
Nitya. Good evening to one and all. On behalf of Department of Chemistry, I take great privilege in welcoming you all for today's program on nanomaterials in antimicrobial photodynamic therapy and biomedical applications. First of all, I thank and welcome our founder chancellor, Thiru A.C. Shanmugam, and President Engineer A.C.S. Arun Kumar, for the opportunity given to us in conducting this program. I welcome our chairman, vice chancellor, provost, and registrars of our institution who are the pillars of our growth. I welcome our joint registrars, Phase II, Dr. S. Ramalingam, Dean Phase II, Dr. S. Manivannan, Deputy Dean Academics, Dr. A.R. Arunachalam, Deputy Dean Administration, Mr. K. Sendil Kumar, Dean Chemistry, Dr. Uday Kala, and Head of the Department, Dr. S. Vadiyami, for being with us and leading all the way. I'm very happy here to welcome our today's resource person, Dr. Busi Siddhartha, a versatile speaker who has accepted our invitation and joined with us today to share his knowledge with us. Welcome you, sir. Today we have students, research scholars, academicians, and research scholars as online participants. I'm happy to welcome you all. Nevertheless, I welcome all my department fellow staff who are with us both in online and offline in bringing out this webinar successfully. Once again, I welcome you all.
Thank you, ma'am. You are warm welcome. Now, I would like to invite Dr. N.R. Nitya, Assistant Professor in the Department of Chemistry, to introduce our chief guest.
We must feel happy to have Dr. Busi Siddhartha, an eminent personality among us for today's program. I take delight in speaking a few lines on his achievements. Dr. Busi Siddhartha is currently working as Assistant Professor in the Department of Microbiology, Pondicherry University. His area of specialization are bacterial quorum sensing, drug delivery, discovery, microbial biocatalysis, and microbial metabolomics. He was awarded CSIR Net Lectureship by CSIR in the year 2003 and 2004. He has qualified GATE conducted by IIT in the year 2002 and has secured 92.6 percentile. As regards publications, he has published more than 85 research papers in international journals and 37 book chapters. He has another feather on his crown by exhibiting himself as one of the editors for the books published in the year 2020. He has completed two research projects funded by SERB and UGC for the total amount of 30 lakhs. He is currently working as a principal investigator for a project funded by SERB for the amount of 38 lakhs. We are happy to invite such an eminent personality for this national level webinar to deliver his valuable speech on nanomaterials in antimicrobial photodynamic therapy and biomedical applications. We welcome you, sir.
Thank you, madam, for your brief introduction. Good afternoon to all the participants and organizers. It's a post-lunch session and it's a little long session. I would like to disseminate some knowledge about photodynamic therapy, how this can be employed, especially to kill the bacteria, multi-drug resistant bacteria. So I have taken two examples. One is based on the CNT, carbon nanotubes, and one is based on the other nanomaterial, that is Mesoporous Silica. So I would like to share my PPT and continue with my lecture. Give me 1 minute of time. So today's talk is around the nanomaterials in antimicrobial photodynamic therapy and their biomedical applications. So it is divided into two chapters. First chapter, I will cover most of the nanomaterials we prepared in our laboratory using multi-wall carbon nanotubes or CNTs. We worked their efficacy against Pseudomonas aeruginosa and Staphylococcus aureus using different photosensitizers. So what is the major problem with the bacteria? Why we have to concern with the bacteria or the microorganisms? Even though this photodynamic therapy nowadays, from bench to the bedside, it has been become one of the prominent cure for dentistry, for cancer therapy and all. Still, lot of research is going on to make it more efficient. So we want to work with the same methodology of photodynamic therapy using a light and a photosensitizer to kill the bacteria. So what are the challenges we are facing? We are facing the challenges from influenza, flu, or tuberculosis, and E. coli, or most of the immunodeficiency virus, now even SARS-CoV-19. So all these diseases are infectious diseases and is one of the main problems worldwide. Therefore, antibiotics are prescribed and we are using these antibiotics in a whole spectrum in a modern medicine. But indiscriminate use of all these antibiotics leads to the emergence of drug resistance. This drug resistance cannot be cured that easily. It results in the prolonged illness, disability, and death. So we came across so many pathogens, whether they are multi-drug resistant or pan-drug resistant, and these are related infections may cause and becomes emerging threat to the public health worldwide. These infections are associated with approximately 2-fold higher death rate compared to the normal because they have a tendency to form biofilm and they will harbor antibiotic resistant genes. This will lead to the resistance and persistence of the infection. So what is photodynamic therapy? So how this is evolved is one of the strategy against infectious diseases. So antimicrobial photodynamic therapy, in a short term we can call it as APDT, is a very promising method for combating bacterial infection, even for resistant strains also, because we are not looking for any target, we are looking for the entire metabolism, either carbohydrate, protein, cell components, etc. APDT involves the use of harmless visible light, like with the different wavelength or a stretch of visible light, combined with a light sensitive dye, which can excite and come down, and this energy can be transferred to split the oxygen molecule to produce reactive oxygen species. So this light sensitive dye is called photosensitizer, and an oxygen present in and around the cell is required. So fortunately, no resistance to photodynamic destruction has been reported to be acquired by the bacteria. because the reactive oxygen species that is produced, this is not specific to one target. It will have a different mode of actions. It has been proposed that the ROS or reactive oxygen species generated by the light activated photosynthesizer can trigger microbial killing and cell damage via three mechanisms. Either damage to the cell membrane, or in case of virus, it will be viral envelope, inactivation of viral enzymes of the proteins, and damage to the DNA. So there are different mechanisms of photodynamic therapy out there. Widely they are classified into type 1 and type 2. Okay, this is the pictorial representation of type 1 and type 2 photodynamic therapy. In photodynamic therapy type 1, okay, pathway, charge is transferred to a substrate or to a molecular oxygen generating reactive oxygen species like hydrogen peroxide and hydrogen oxygen radicals, like superoxides, ions, and free hydroxyl radicals. Then they will generate the reactive oxygen species. In type 2, energy is transferred directly to the molecular oxygen, where the highly reactive singlet oxygen is generates, and it can kill the bacteria. So you can see here in the pictorial representation both type 1 and type 2 and how they will act the cross generation microbial cell death. So normally this photosensitizer delivery and nanomedicine, why we opted the nano? Because one of the most important problem in photodynamic therapy
Hello,
is
sorry.
optimizing the delivery of photosensitizer.
Hello,
Most
sir.
of the photos, yeah, yes, madam.
Sir, could you please share the slideshow mode, sir?
Yeah, it is in slideshow only, madam.
No, we cannot see, sir.
You
It's
are
in the
unable to
first
see the
slide.
slide?
Yes, sir. It's in the still in the first slide itself.
No, no, madam. It is there in the slideshow. Can you stop and present again?
Yes, sir. Yes, sir. Sure, sir.
So now it is in slide share mode. Now it is in full length slide, madam.
It's not in slideshow mode, but it's in the PPT itself, sir. Could you just move on the slide there itself and go ahead, sir?
Okay. So
Or
can I
you,
call it, because I'm in slide share mode, madam, in sharing window only.
okay. Okay, sir. Please go ahead, sir. It's in the
Okay.
seventh slide, right?
Yeah, in type 2 pathway, energy is transferred directly to the molecular oxygen. So why we used nanomedicine? One of the most important problems in the photodynamic therapy is delivery of photosensitizer. Okay, the most photosensitizers are insoluble in water, are insoluble in a given material, and they tend to aggregate. This aggregation may leads to the very poor efficacy of the photosensitizer. So the unique physical and chemical properties of nanoparticles, like their small size, high surface to volume ratio, can make this photosensitizer to deliver better, or they can be used as a carrier. So allows this technology to surpass the barrier against the access to biological molecules and systems. And second one, some of the photosensitizers like fullerenes, nanoparticles like fullerenes or CNTs or Mesoporous Silica, by virtue of their nature, they have the capacity to kill the bacteria. So we can combine a photosensitizer and a nanomaterial that can lead to a better activity. Nanoparticles have been recently used in APDT to enhance the effectiveness of the treatment. So what are the advantages of nanoparticles and photosensitizer? Large concentration of photosensitizer for the production of lethal ROS. If the PS is not soluble in water, encapsulation can be used to enhance the solubility or slow release, stopping the PS from dimerizing and dimerization so that they will be in a free state and their effectiveness can be preserved. Reduce stability of the target cell to pump out of the PS, hence reducing the possibility of multi-destruction, and selectivity of treatment by localized delivery agent. So we can use a treatment by localized delivery agent like in diagnostics. In the present study, carbon nanotubes are emerging as an attractive scaffold. So we use carbon nanotubes. Okay, the brief introduction of carbon nanotubes are they're long, hollow cylindrical nanostructures wrapped up by graphene sheets. One of the basic carbon nanotubes are classified into either multi-walled or single-walled, okay, based on their number of walls. CNTs have the unique potential application in various fields because of their unique physical, electrical, and mechanical properties, such as high aspect ratio, large surface area relative to their molecular weight, and they have an property, okay, mechanical property due to the sp2 carbon to carbon carbon bonding. So structurally, this multi-walled carbon nanotubes consists of multiple layers of graphite, okay, superimposed and rolled in on themselves to form a tubular shape. So this tubular shape we can able to use as a carrier of many of the photosynthesizers inside. This makes them to be promising potential for delivery of the drugs, peptides, nucleic acids, and so on. CNTs, antimicrobial activity, they have, okay, they can able to kill the bacteria by cell membrane perturbation or direct oxidation of cell components or secondary oxidation of lipids, lipid bilayer, etc. So these single-walled CNTs have been demonstrated to exhibit antimicrobial activity stronger than that of the multi-walled carbon nanotubes. So coming to in this first chapter, I would like to introduce even the photosynthesis what we studied, that is toluidine blue and the malachite green. This toluidine blue is a cationic photosynthesizer or a dye. It is absorption band at 630 nanometers. It has photodestruction activity towards the both gram-negative and gram-positive bacteria. So it is widely studied for photodynamic therapy. So malachite green is a cationic dye and it belongs to the triaryl methane dye. It is an malachite green absorption is around 670 nanometers. The antimicrobial photoinactivation ability of this malachite green is more active on gram-positive bacterial species has been widely studied. So these are already studied, but our hypothesis is when I combine this malachite green or toluidine blue with multi-walled carbon nanotubes, how we can able to increase the efficacy. So the objectives of my study are synthesis and functionalization of carbon nanotubes and characterization of synthesized carbon nanotubes, conjugation of photosynthesizer with synthesized carbon nanotube, then followed by characterization of this photosynthesizer and the CNT conjugate, and in vitro uptake and release. These are very, very important parameters. How much uptake is there and how much release of the photosynthesizer into the system. Okay, we are studied. And photodynamic inactivation of planktonic cells, photodynamic inactivation of biofilms, and what is the mechanism of action, and the statistical analysis. So out of this entire study, we came to know, okay, the effectiveness and enhanced photodynamic inactivation of Pseudomonas aeruginosa and S. aureus, these two are the pathogens, was achieved by TB-MgCNT conjugate. The antimicrobial and antibiofilm activity of TB-MgCNT against test bacteria was due to the production of reactive oxygen species. We established it. And the synthesized conjugates can be used in the treatment of infections. So first study is toluidine blue doped carbon nanotubes. So spectral calculations and experimental parameters for this are, we used a diode laser with a 670 nanometers wavelength with a peak wavelength of 630 nanometers. Operation mode is continuous mode with a radiant power of 125. Polarization is random. Okay, the number of treatments we did is one-time treatment, and the eradication area is 0.385, which is we used the 96 culturates to for an irradiated area. So coming to the results of this study, so we first used CNT, and as in the graph in the A, we used CNT, okay, and TB, and we have known their absorption spectra. So 280 for CNT and 360 we graphed for the TB. So whereas when we conjugated TB-CNT, we find both the peaks, that is at 280 and 630 are near to 630. It indicates that my conjugate is having both CNT and TB. This second B is an representation of an TEM for TB-CNT. You can see all the CNTs in a very long thread-like structures. So then we want to know the functional groups, whether conjugation happened properly or not. So we graphed using FTR spectra. In FTR spectra, we have taken the CNT. In CNT, we came to know 3451, 1544, and 1458 and 1073. We identified some of the functional groups. Whereas in TB, we came to know that there are functional groups as 3267, 1613, 1535, and 626 like that. And when we have a conjugate, we have both the functional groups of CNT and TB graphed between 3442, 3922, 2333, 1592, and 1060. This U visible in accordance with the FTAR confirms that, okay, both we are having a conjugation. Further, we used Raman intensity, Raman spectroscopy. In CNT, we identified 1312 and 16032 two peaks, and 1448, 1032, 1392, and 1623 for the TB. Whereas in a conjugate, we identified 1309 that of TB, 1600 that is that of both the TB and the CNT, and other graphs also, other wave numbers also. This indicates these results confirms that okay, TB CNT conjugation has been happened. So then we want to go with the excitation and emission spectra. So when we taken the CNT excitation is at 375 and emission was identified at 290. For TB, this was identified at 670, excitation and emission at 290. So our hypothesis is the excitation and emission for TB CNT should be the both of both CNT and TB. So when we studied TB CNT excitation and emission, we find 657 and for emission and excitation of 625 for TB in TB CNT conjugate, and in CNT 361 and 273. And this indicates that okay, the TB CNT conjugate is having both TB and CNT together. So then we want to know how much compound has been loaded or entrapped. So this is called loading capacity or entrapment efficacy. So loading capacity we identified as 12.2124 and the entrapment efficacy as 48%. So these percentages are calculated according to the formula given below. So then we studied the laser exposure. The radiant exposure was calculated. Radiant exposure was calculated using the power density and exposure time. All the experiments are conducted in triplicate using this laser light photosensitizer and with the CNT, and all the results are expressed with the significance, with their significance. So this one, we want to know all the time how the CNT is working, how TB is working, and how TB CNT conjugate is working. Not only that, we want to even study how this CNT is working in the dark condition, then how the CNT is working in the light condition, and also how TB is working in the dark and light condition. So we want to know light and dark conditions, how there is an influence. So percentage uptake of TB was identified. Okay, this is the first one. A is percentage release. Okay, in percentage release, we identified almost 99% release of the TB over 180 minutes. And the percentage uptake by the bacteria, not only the release, the bacteria like CNT and TB, when I check the TB only alone, TB percentage uptake is around 40% at 180 minutes, whereas for TB CNT, here TB has been uptaken by around 58% over the period of 180 minutes. So later we want to know the uptake of the TB CNT using the one more bacterial isolate. There also we identified almost 60% of uptake. So then we want to study how this bacteria is able to kill the planktonic cells. This is the D is a representation of killing the bacterial cell. CNT, okay, in light, okay, L minus means in the dark, L plus is with the light. With the light means laser. So when there is a dark, the CNT, okay, has been reduced the number of cells very less compared to in the light. In TB also, there is a very less reduction is there in the dark and compared to the light. But when we have an TB-CNT conjugate, in the dark, there is a very less reduction, around one-fold reduction is there in the bacterial cell, but it has been enhanced to the four-fold reduction when it has been exposed to the light. This indicates that compared to the individual and compared to the dark mode of action, when we are giving a laser, TB-CNT can able to kill or destruct the bacterial cell effectively. So then we want to know how this is happening. Whether the TB-CNT is inducing any ROS. Yes, we want to study that ROS generation by DCF fluorescent intensity. So here also we adopted the same method. Okay, L minus is in dark and L plus is with the light. So we have taken a control of CNT, TB in the dark, and the TB-CNT and TB and CNT in the light condition. So when we see the ROS generation or DCFDF fluorescence, which is directly proportional to the ROS generation, you can see highest ROS generation in the TB-CNT. That means more amount of ROS is generating, that is killing the bacteria. So then we want to know how this bacteria is having effect on the protein leakage, means how the cell's membrane has been damaged and protein is leaking out of the cell. So we want to quantify the supernatant after damage and to see how much protein is leakage. If more protein is leakage, more cell damage you can able to imagine. So then when we use the CNT and the TB in the dark, there is very less amount of protein has been leakage. When we use the TB-CNT in the light, the protein leakage has been almost 60 to 80%. So that indicates that cell damage has been occurred extensively. So then we want to study lipid peroxidation. Lipid peroxidation is also will be calculated by MDA equivalence. When we study the lipid peroxidation, there will be very less lipid peroxidation we saw with the control CNT, TB in the dark condition, and when we use the TB CNT in the light or exposed to the laser, there is a more destruction of the bacterial cells, and that is equated with the MDA equivalence of lipid peroxidation. So this is one more. This is about the biofilm characters, because ultimately the bacteria will lead to the biofilm formation, and this tends to antibiotic resistance. So we want to study both planktonic cells and biofilm cells. So this study is A represents the biofilm percentage. You can see a drastic increase in the biofilm destruction or biofilm inhibition when TBCNT is exposed to the laser. When TBCNT is alone, very less reduction is there, and TB alone or CNT alone in the light and dark condition, there is a very less destruction of the bacterial biofilm. This second picture is represents the, in the B, it represents the percentage of TTC or cell viability. So cell viability has been decreased almost 80% or 70% to 80% in TBCNT exposed to the light compared to the TB or CNT individually in the light or when exposed to the laser. So this C is a representation of reduction of EPS production. EPS is an exopolysaccharide matrix. In biofilms are covered with this exopolysaccharide matrix, and that leads to the antibiotic resistance. So this exopolysaccharide will not allow any of the bacteria to get into the cell. Okay, so this APS, how much reduced we want to know. So there is a more reduction of exopolysaccharide was observed in TBCNT when exposed to the light when compared to the CNT or TB without light or with light. So then we want to visualize the effect. When we use the confocal laser scanning microscopic analysis against Pseudomonas and Staphylococcus aureus, so we can see in the control all this green color is in live bacterial cells with a dense bacterial biofilm. Whereas in CNT, okay, alone with the dark or TB alone in the dark, very few cells were dead. When we are using TBCNT in the dark, also you can see the pink color or red color cells. There are very less are there. That is the representation of dead cells. When we expose it to the light, okay, either CNT and TB, you can have a photo destruction with photosensitizer. But when you see the TBCNT, this is H, this is indication of more dead cells with an orange color or red color here. In case of SRUs also, in control you have an profound growth of biofilm, whereas if you take TBCNT exposed to the light, you have a more photo destruction can be observed. So this is an indication that we came to know that the TBCNT is a more active compared to its individual drugs. So malachite green doped carbon nanotubes we would like to study later on. So first one is TB, then MG. So we studied that malachite green loading capacity is 10% and the entrapment efficacy is 82%. So here also we graphed the malachite green. Malachite green is having a peak at 618 in UV visible spectrum, and a CNT is having 270. But when we have an MG-CNT conjugate, we find both the peaks, indicating that they have been encapsulated. This is a TEM picture indicating that the CNT structures and all. So similar to that of the previous study, we graphed the FTIR for CNT, MG alone and MG-CNT. So here we graphed that 3404 and 2362, 455 are very important regions. Whereas for MG, okay, we identified that 3440, 1563 and 1172 are important. So in an MG-CNT conjugate, you can see the same functional groups at 3422, 1641 and 1095, indicating that all these compounds has been conjugated. So again, we studied the photoemission excitation spectra for MG-CNT, where we find 655 intensity of emission and the excitation of 630. So this is a release of malachite green and uptake of malachite green. So when we use the percentage release of malachite green is around 26 or 28 when exposure to 180 minutes. And coming to the percentage uptake by the bacteria, the percentage uptake of the bacteria when we use the MG, it is only restricted to around 19%. But when you use the MG-CNT, the percentage uptake is a slow release. So the percentage uptake also increased around 30%. So percentage then we want to study how this photodynamic therapy is working for the MG-CNT. So as that of the previous studies, CNT alone or MG alone in the light condition have very less effect. And when we use the MG-CNT with laser exposure, the fold increase in the log reduction is increased to 4-fold or more than that. So then we would like to study reactive oxygen species, protein leakage, and MDA equivalence, that is lipid peroxidation and biofilm inhibition. In all these ABCD graphs, okay, you can see the control, okay, L minus means that there is a dark, L plus is a light laser exposure. When we use the laser exposure for CNT and MG, the photo destruction is very less. Okay, but when we use the MG CNT, okay, with the light, the amount of photo destruction is very high. This is evidenced by Ross generation. This is also evidenced by the protein leakage and also evidenced by the lipid peroxidation and biofilm inhibition. So it is equivalent both in case of Pseudomonas and as well as SRES. So then we want to see the number of cell viability, okay, that is a reduced cell viability on against Pseudomonas and SRES, and also significant reduction in the EPS production. So in this case also, okay, the light and dark, in a light induced, will be photo destruction in the little photo destruction in MG and MG CNT, but when the conjugate has taken, there will be more photo destruction. And similar way, exopolysaccharide production has been hampered or completely reduced when we have an MgCNT exposed to the laser. This is a confocal laser scanning microscopic analysis. We would like to visualize. A is a control. Okay, in A we can see more amount of green cells, that is the live cells. Okay, whereas the G is nothing but an Mg when we expose it to the laser. H is MgCNT, that is a conjugate, nanoconjugate. When we expose it to the laser, there is a more photo destruction. You can see more orange and red color cells. Similar way, the B is representation of SRES. In here also, in BA, okay, here you can see more amount of green cells which are related to the live cells, whereas in H, that is a MgCNT, with the laser exposure, there is a more photo destruction. So coming to the conclusion of this particular chapter, the field of nanotechnology is now known to be an integral part of medicine with a wide application in drug delivery, drug discovery, and disease diagnosis. One of the emerging drug delivery agent is carbon nanotubes. Recently, they gained more interest in antimicrobial therapy as well as antimicrobial therapy and drug delivery. We have summarized the efficacy of photoactivated TB or TBMG or MgCNT conjugated for antimicrobial therapy against Pseudomonas and SRES. So the ability of TBCNT or MDCNT conjugated to a conjugated have a potential to inactivate the bacteria more in an effective manner. The conjugate was successfully employed for the reduction of planktonic cells as well as biofilms. In future, these conjugates can be used as a potential alternative for the treatment of localized infections or surface colonization of bacteria in hospital instruments. These are the references what we followed for this study, and these are the publications we made out of this study. One is in photodiagnosis and photodynamic therapy using carbon nanotubes and toluidine blue, and the other one is photochemical and photobiological sciences, synthesis of this CNT and their conjugation with methylene blue against E. coli and SRES. Other publication is related to photodiagnosis and photodynamic therapy. This is Rose Bengal. We encapsulated with multi-wall CNT and against E. coli. And other one is International Journal of Nanomedicine. Malachite green we conjugated with the multi-wall carbon nanotubes, and we studied its activity against planktonic and biofilms of Pseudomonas and Staphylococcus aureus. So then we have an interest on Mesoporous Silica also. Mesoporous Silica also one of the important material nowadays gained a wide application and interest among the researchers. So we want to study this Mesoporous Silica, how this can able to be used as a nanocarrier in photodynamic therapy against Candida albicans. So the second chapter is related to the inactivation of fungal biofilms using amino functionalized Mesoporous Silica, Rose Bengal conjugate against Candida albicans. So again, this when I talk about the fungal diseases, very recently one of the report last week, okay, it says that in Andaman, Candida auris is being isolated, and Candida auris is a multi-drug resistant, and the WHO is estimating in the coming 10 years there will be more than 10 lakh deaths because of this Candida auris. It is a multi-drug resistant. Identification of them is very difficult. So similar to the top that many candida species are available. They are clinically significant and they have an resistance pattern. Okay, this leads to the more mortality rates. In 2011, WHO declared comeback drug resistance, no action today, no cure tomorrow. Means we are running out of antibiotics. Almost all the microbes are getting resistance against many of the antibiotics. So that leads to the emergence of multi-drug resistant, pan-drug resistant, and extended spectrum resistant antibiotics antibiotic resistant strains worldwide. So this antimicrobial resistance poses a major threat to the patients and their treatment. It leads to the morbidity and mortality, increased hospital stay, and a severe economic loss to the patient and the nation. The rapidly increasing emergence of antibiotic resistance among pathogenic microbes may bring more than thousands of deaths over the past 50 years, termed as the antibiotic era. So one of the major cause of this AMR is the ability of microorganisms to form biofilms, either it's in the case of bacteria or in the case of fungi. Biofilms are microbial communities. They're attached to the surface and encased in an extracellular matrix of microbial origin. Biofilms are everywhere and can be developed on virtually every natural or man-made surface, either abiotic or biotic surface. According to NIH, it is estimated that about 80% of human infections are associated with the biofilm-forming microorganisms. So biofilms are having clinical importance. Bacterial biofilms and their role in disease has been studied intensively, and in recent years, now we are considering them to mitigate these biofilms, especially the bacterial and fungal biofilms. So coming to a brief introduction about my model organism, Candida albicans, the pathogen to kill. The Candida albicans is an opportunistic fungus, produces both superficial and systemic infection. Especially in immunocompromised individuals, you can see severe infections due to this C. albicans. The ability of Candida to change from yeast to filamentous form is a crucial process for colonization and virulence that leads to the subsequent formation of biofilm on the variety of surfaces, including host tissue, wounds, or medical devices. Candidemia or candidiasis has been a leading type of nosocomial infection with a high morbidity and mortality, which accounts around 20 to 40%. The biofilms are resistant to a range of antifungal agents currently in clinical use, including amphotericin B, fluconazole, and there appears to be a multi-resistant mechanism. So these are the steps involved in the biofilm formation. The bacteria, it will be attached to an substrate, they will multiply, they will produce a kind of signaling molecules that is called as a quorum sensing, and they will multiply. After multiplication, they will produce a lot of exopolymeric substances like eDNA, alginate, exopolysaccharide, granulolipids. Because of that, they will be encased. This is called biofilm matrix. In this biofilm matrix, no antibiotic can able to enter. So once they are having a matured biofilm, slowly the superficial cells will be ruptured and they will be dispersed, and again they will fall onto the other surface to start a new microcolony or the biofilm formation. So here we want to know the photosensitizers in this study also. Here we use the rose bengal. The photosensitizer is a non-toxic in the dark, especially towards mammalian cells. So they should be good quantum yield of ROS and have a molecular absorption coefficient at a wavelength where high penetration of tissue is good. PS should show selectivity for microbial cells over mammalian cells. It should not be toxic to the mammalian cells. PS should have cationic charges ideal provided by quaternary nitrogen atoms or basic amino acids. This is the structure of rose bengal, and in this study we are using rose bengal with an absorption property of 540 nanometers. So coming to the Mesoporous silica, among the nano vehicles used in the nanomedicine, Mesoporous silica is gaining more interest in APDT because of their biocompatibility, high pH loading capacity, and ease of surface functionalization. You can do either carboxy functionalization or amino functionalization. The unique properties of Mesoporous silica, such as they have a controlled particle size, porosity, so that we can able to dope any drug into that, and the morphology and high chemical stability makes this Mesoporous silica nanoparticles highly attractive drug carrier for diagnostic catalytic property or maybe for separation and sensing applications. Rapid internalization by animal and plant cells without causing any cytotoxicity inside the body is one of the important features of this Mesoporous silica. It will not cause any deleterious effects. The biological application of Mesoporous Silica nanomaterial includes imaging and diagnostic agent, specificity, dispersibility, and capability to load or deliver high concentrations of photosensitizers. We can able to go for a therapy or diagnosis. These are the objectives of this particular study. We are functionalizing the Mesoporous Silica, that is amino functionalization. We would like to characterize the Mesoporous Silica, whether conjugation happened or not, or functionalization happened. Then Mesoporous Silica PS conjugate is characterized, and we studied the in vitro uptake and release of this photosensitizer into the fungal system, and then photodestruction of these planktonic cells and biofilms using this nanoconjugate, study mechanism of action and statistical analysis. So coming to the results, we performed similar to that of the previous studies, FTR analysis for Mesoporous Silica and after functionalization. So we used Mesoporous Silica from Sigma Aldrich, and we used 3-amino profile trimethoxysaline, that is a APTMS, 95% soluble in the ethanol, so that it can become amino functionalized. So in MSN, we came to know that 804, 964, 1092, 1629, 3459 are the important functional groups. Whereas in MSN NH2 amino functionalized, we find even amino groups, that is 1642, 2843, 2880, 2953, 3436, and apart from that of the silica or oxygen and hydrogen bonds, that is similar to that of MSN. So conjugation of photosensitizer with functionalized MSN. So we have first step of functionalization, then second step is conjugation. So conjugation of functionalized Mesoporous Silica with rose bengal was performed with a 5:1 ratio using ethanol as a solvent. After conjugation, the color of the compound obtained was a pink colored powder. So further, it was subjected for the characterization using UV visible spectrophotometer, photoluminescence, excitation and emission spectra, and FTIR. So this is the UV visible spectra, where in UV visible spectra for RB, okay, that is a rose bengal, we find a peak at 540, and a similar peak was observed in the MSN RB also at 540. This indicates that the compound has been functionalized or is conjugated. In photoluminescence analysis, we identified photoexcitation and photoemission. The photoexcitation and emission is similar to that of the individual molecule, and also is similar, the excitation and emission was observed in MSN RB conjugate. Okay, the bottom one is this is MSN RB excitation and emission. This is RB excitation and emission. So FTI analysis was performed for MSN RB separately and the conjugate MSN RB. So here we identified there are different functional groups at 469, 800, 591, 1090, 1348, 1642, and so on for amino group and silica group for MSN with amino functionalized. For RB, rose bengal, we identified the functional groups at 534, 568, 615, 956, 1455, 1548, 1611. So similar kind of functional groups were identified for silica at 467, 801, 953, 1094. For RB also, similar groups of RB, that is 1450, 1547, 3632 was identified, which indicates that my photosensitizer RB has been encapsulated in the MSN. So loading capacity and entrapment efficacy were identified. Loading capacity was identified by the formula weight of the loaded dye by weight of the dye loaded MSN into 100. Entrapment efficacy was identified by the weight of the loaded dye by the weight of the dye in field. Okay, then we came to know MSN was loading capacity of MSN is 11%, whereas entrapment efficacy is 58%. So then we want to see the release. Loading is not important. Along with the loading, release also should be there. If it is tightly loaded or maybe packed with a more bonding, strong bonding, the release will be weak. So we studied the release also. The release of the dye started from 30 minutes. It went up to the third hour. In third hour, we identified that there is more than 60% of release was there. So apart from release, we studied the uptake. So at different intervals like 30 minutes, 1 hour, 1.5 hours, and 2 hours, when we studied the uptake, RB alone, the uptake at second hour is 36%, where the uptake has been increased at 30 minutes when you used RB conjugated with the MSN. So here, after MSN, we want to see the RB-MSN conjugate, how it is working. So in both the dark and light, we use the laser. Okay, in this laser also, when we expose it with the light and dark condition, the green bars represents the dark condition and red bars represents with the light, exposure to the light. MSN alone is having very small effect, whereas RB is a little more effect. But when we combine the MSN RB with the light condition, the photo destruction is more than 80% and significant compared to that of the individual RB or MSN. It indicates that MSN RB can be an effective material compared to the MSN or RB alone in a light-induced condition. So further, it is validated using spot assay. You can see here, C is a control. The clear bacterial spot at the colony you can see here, even in the dark and light. Okay, whereas when MSN is exposed to this bacteria, you can see still the spots are there. RB, in the dark also you have a spot. When we have a light, there is a little photo destruction was observed. When we used MS and RB, the dark condition, there is a little photo destruction and more photo destruction was observed when we induced the light. So then we want to study the cellular loss generation and protein leakage. So when we expose this material with the bacteria, so in fungi, C. albicans, there is a more loss generation was observed. So this is the intensity of loss in the dark and light condition. Blue bars are represented with the light. So you can see blue bars when there is a culture, very less, and when only MSN, there is a little destruction, RB also little destruction. But when we expose it with the light, MS and RB, there is a more photo destruction was observed and more cellular loss generation was observed. So coming to the cytoplasmic leakage in this figure, here also you can see compared to the MSN and RB in the dark and light condition, the light condition of MS and RB has been more photo destruction when treated to the C. albicans. So this is the DNA damage. This figure indicates, okay, these are the different ladders we used for control, MSN, RB. Okay, the last one is MSN, RB, and similar way. So there is a more bacterial DNA damage was observed in the lane where MSN, RB is treated with the C. albicans. And lipid peroxidation also, when we take the MDA equivalents or lipid peroxidation, there is a more lipid peroxidation you can see when there is a light-induced death with the MSN RB. When we use MSN or RB, in the dark condition, there is a very little photo destruction is there, or little destruction of the bacteria. When we use the light condition, also the similar way, but more photo destruction, more bacterial death was observed in photodynamic therapy when MSN RB is coupled with a light source and given to the Candida albicans. So this is for the biofilm inhibition and cell viability. Biofilm inhibition in the dark and light also we studied, where in the light induced MSNRB can able to destruct more C. albicans biofilm compared to that of the control or MSN itself or RB. Similarly, cell viability by TTT assay, okay, we studied. Here we can able to see the number of live cells. Reduction in the live cells has been reduced to 80% or more than 80% live cells has been reduced. Okay, even MSNRB has been used in the light condition. So this is a EPS quantification. For C. albicans also we studied for EPS quantification in the dark and light when MSN, RB or MSN, RB is in the dark condition or light induced. In the light, when we are exposed to the light MSN, RB, there is a significant reduction in the bacterial EPS compared to that of the MSN or RB exposed in the light or in a dark. This is a confocal laser scanning microscope. Here, okay, A is in control. Okay, when we take other images, this H is MSN RB and the light exposure. This is RB alone. Okay, this indicates that there is a more red color cells, more photo destruction when MSN RB is given to the C. albican system and exposed to the laser. Coming to the conclusions from this study, C. albicans is the most common superficial fungal infection in humans and an effective therapy to kill the cells and reduce their biofilm associated virulence is required. Fluconazole or azole drugs are widely used, but they got resistance and also these biofilms makes them to be ineffective. The present study emphasizes the prepared nanocomposite, that is MSN rose bengal RB, significantly enhance the antimicrobial photodynamic therapy against planktonic cells and biofilms of Candida albicans. Antimicrobial photodynamic therapy of this MSNRB nanoconjugate is more efficient against C. albicans due to their PS binding efficacy, enhanced RYS production, protein leakage, DNA damage, lipid peroxidation. That's a multitude of target and a significant biofilm reduction than that of the pre. Hence, nanocomposite of present study can be used to eradicate the growth and virulence of C. albicans significantly. These are the references we followed for this study, and these are the publications we used MSN extensively with different photosensitizers. The first one is published in PeerJ, where antimicrobial photodynamic therapy on Staphylococcus aureus and E. coli using malachite green and Mesoporous Silica. Okay, this one, the other paper is in biofouling and antimicrobial photodynamic therapy of toluidine blue encapsulated Mesoporous Silica against Pseudomonas and Staphylococcus aureus. And this is a book chapter on application of carbon-based nanomaterials for antimicrobial photodynamic therapy, where we extensively given the information about CNT and MSN. And this is a journal of Scientific African. Here, the present study, what I explained, antimicrobial photodynamic therapy of MSN with rose Bengal conjugate against the Candida albicans. Okay, thank you with this information.
Thank you, sir. Thank you very much for your wonderful talk. You had a great work in the research area for synthesis of carbon nanotubes, mesoporous silica nanoparticles, the characterization, and excellent biomedical applications. So we came to know so many things from this section. We would like to give you an appreciation certificate from our organization as a representative of our gratitude. So please accept it, sir.
Thank you.
The feedback form will be available in the YouTube chat box. So I request all the participants to fill the form and submit before 5 p.m. Later, the form will be deactivated and the e-certificate for the participants will be sent to your registered email ID immediately. Now, I would like to call my colleague Mrs. Jeevarathinam to propose vote of thanks.
Jiva ma'am, please unmute yourself, ma'am.
Good afternoon to everyone. I am Jeevarathinam, Assistant Professor, Department of Chemistry. I take this opportunity to propose a vote of thanks on behalf of the organizing team. My heartfelt gratitude to our Honorable Chancellor 3AC Sanmugam, Honorable President Engineer A.C.S. Arun Kumar, Vice Chancellor, Provost, the Registrar and all the Joint Registrars, Dean Dr. S. Manivanan Phase II, who encouraged and motivated us to organize the program. And also I would like to thank Deputy Dean Academic Dr. A.R. Arunachalam and Deputy Dean Administration Mr. K. Sendil Kumar and Dean Chemistry Dr. P. Udayakala and HOD Dr. S. Valliyamai for their continuous encouragement and support. On behalf of my university, my special and heartfelt thanks to our today's speaker, Dr. Busi Siddhardha, Assistant Professor, Department of Microbiology, Pondicherry University, who delivered his presentation in a well-explained, useful and interesting manner. Thank you, sir. I extend my sincere thanks to all the participants for spending your valuable time and listening us. I would also like to thank all the panel members, Dr. L. Ramapriya, Dr. R. Nitya, Dr. D. Kavita and Mr. Ganesh Babu for technical support. Once again, I would like to thank all the participants for making this event in a success manner. Thank you all.
Dr.M.G.R Educational and Research Institute, University, Madurai, Chennai.